Hi everyone, and welcome to our module on ammonia. Unlike fats and carbohydrates, there is no storage form for amino acids or proteins, so any excess amino acids or unused amino acids are broken down by the body. The way the body does this is by removing the amino group from the amino acid to create an ammonia molecule and a structure called an alpha keto acid. If we look at the left side of the screen here at the bottom, this is the generic structure of an amino acid. We have an amino group attached to an alpha carbon and then a carboxylic acid group, and there is a side chain that is different for each amino acid represented by the R. This can be broken down into ammonia by removal of the amine group, and then we will have a structure called a alpha keto acid. This is called an alpha keto acid because there is a keto group attached to the alpha carbon, and then there is a carboxylic acid group here. And this is the generic way by which all amino acids are metabolized in cells in the body. The problem with this metabolic pathway is that ammonia is toxic to the body, specifically toxic to the brain. We'll talk more about that later. So it needs to be transferred to the liver in a non-toxic structure, and then once it gets to the liver is going to convert it to urea, which is a non-toxic molecule that can be excreted by the kidneys. The usual first step in metabolism of amino acids is removal of the nitrogen group by a process called transamination. And usually the amino group from the amino acid is passed to alpha ketoglutarate, and therefore the alpha ketoglutarate becomes a molecule of glutamate. If you look at the bottom of the screen here, on the bottom left is a generic amino acid structure. At the top left is the structure of alpha ketoglutarate, which you may know is a TCA cycle intermediate, and it's found in cells throughout the body. What happens in the transamination reaction is this amino group here will be transferred to where this oxygen is attached on alpha ketoglutarate. The resulting structure is a molecule of the amino acid glutamate, which now has the ammonia group attached here, and the ammonia has been removed from the amino acid, and therefore we have an alpha keto acid structure. There are enzymes called aminotransferases for many of the different amino acids in the body, and all of them do the same thing. They transfer nitrogen from a specific amino acid to create a molecule of glutamate. All of them require vitamin B6 as a cofactor, and there are two aminotransferases that you probably know very well from liver function tests. Two of the liver function tests are measurement of levels of alanine aminotransferase, or ALT, and aspartate aminotransferase, or AST. And these are two of the many enzymes that participate in removal of the ammonia groups from amino acids. Just to make this very clear, let's take a look at one of the aminotransferase reactions. Let's look at the one that pertains to the amino acid aspartate. This aminotransferase reaction is catalyzed by the enzyme aspartate aminotransferase, or AST, and it takes the ammonia from aspartate and passes it to alpha ketoglutarate. In doing so, it will create a molecule of glutamate, and it will also create the alpha keto acid of aspartate, which happens to be oxaloacetate, which you may know is a TCA cycle intermediate. So as we have seen, many amino acids pass their nitrogen to glutamate, but what happens to the glutamate? Well, it turns out there are two major methods by which most cells in the body transfer the nitrogen from glutamate to the liver so that the liver can excrete the nitrogen in the form of urea. The first method is glutamine synthesis, and the second method is the alanine cycle. Glutamine is a non-toxic structure that shuttles nitrogen to the liver for excretion. There is an enzyme called glutamine synthetase that is found in most tissues, and it takes all those molecules of glutamate that have been synthesized from breakdown of amino acids, and it converts them into glutamine. To do this, it adds another nitrogen molecule from ammonia, and therefore the glutamine carries two nitrogen molecules back to the liver for further metabolism. In the liver, the glutamine can be converted back into glutamate. There is an enzyme called glutaminase, which will remove an ammonia group and create a molecule of glutamate. There is then an enzyme called glutamate dehydrogenase, which can break down glutamate into alpha ketoglutarate and another ammonia molecule. And then this ammonia molecule, as well as the one created by glutaminase, can both enter the urea cycle, because now we are in liver cells, which is where urea metabolism occurs. An alternative method to glutamine for transfer of nitrogen to the liver that is used by muscles is called the alanine cycle. In muscles, glutamate nitrogen will be passed to alanine, and then the alanine will shuttle it back to the liver. This reaction is catalyzed by the enzyme alanine aminotransferase, or ALT. It will take molecules of glutamate and pass the nitrogen from glutamate to pyruvate and remove the double bonded oxygen from pyruvate. When it does this, it creates a molecule of the amino acid alanine. Alanine has the same structure of pyruvate, except that the ammonia group has replaced the double bonded oxygen. This process will also generate a molecule of alpha ketoglutarate, which can be used by the cells in the TCA cycle. In the liver, the reaction is reversed and alanine is converted back to pyruvate. This is catalyzed by the same enzyme, alanine aminotransferase, or ALT. In this case, it will transfer the amino group to alpha-ketoglutarate. This will regenerate glutamate in the liver and also regenerate pyruvate, which can then enter glycolysis or gluconeogenesis. And just like we saw before, when glutamine was the shuttle for nitrogen, when alanine is the shuttle for nitrogen, the glutamate that is created in the liver can be metabolized by glutamate dehydrogenase into a molecule of alpha-ketoglutarate and an ammonia molecule, which can then enter the urea cycle for further metabolism. So here's a picture of the alanine cycle, the type that you'll find in many textbooks. In muscle cells, the ammonia from amino acid breakdown is passed to glutamate, which transfers it to alanine, and alanine is the shuttle that carries the nitrogen molecules to the liver. In the liver, alanine passes the nitrogen to glutamate, which can then synthesize ammonia, which can then enter the urea cycle for further metabolism. When alanine passes the nitrogen to glutamate, it creates a molecule of pyruvate. Now, via gluconeogenesis, pyruvate can be converted back into glucose, and the glucose can be released by the liver and go back to the muscle cells, where the muscle cells can metabolize the glucose into pyruvate, and thus the cycle can begin again, and that's why it is called the alanine cycle. A clinical correlation of the alanine cycle has to do with rare inborn errors of metabolism called mitochondrial disorders. Babies born with one of these disorders often have deficient metabolism of pyruvate. These can occur with a pyruvate carboxylase deficiency or a pyruvate dehydrogenase deficiency, and in both cases they cannot metabolize pyruvate. As a result, these babies often develop a lactic acidosis but also an elevated alanine level. If we go back a slide, if the level of pyruvate is elevated because of a mitochondrial disorder, that will result in high levels of alanine. That alanine will be transferred to the liver, but the liver cannot metabolize the pyruvate back into glucose because that depends on enzymes in the mitochondria. Therefore, the level of alanine in the plasma can get very high, and elevated levels of alanine are commonly seen in babies with mitochondrial disorders. So now let's talk about the urea cycle. This is a metabolic pathway that occurs only in the liver, and it is the pathway by which the toxic ammonia is converted to the non-toxic structure urea, which can be excreted in the urine. This is the structure of urea on the right side of the screen. It is synthesized from ammonia and CO2. CO2 is what makes up the C double bonded to oxygen in the middle. And then finally, the second nitrogen comes from the amino acid aspartate. The first and second reactions of the urea cycle occur in the mitochondria. The mitochondria is where ammonia is combined with other molecules to begin forming urea. And the first step of the urea cycle is the rate limiting step. In this step, ammonia molecules and carbon dioxide molecules are combined to form a structure called carbamoyl phosphate. This step is catalyzed by an enzyme called carbamoyl phosphate synthetase 1. It involves two molecules of ATP, and the result is a molecule of carbamoyl phosphate, which has the carbon dioxide here, the ammonia here, and one of the phosphates from ATP added on here to the molecule. This first step has a very important activator called N-acetylglutamate. N-acetylglutamate is an allosteric activator of the enzyme carbamoyl phosphate synthetase 1. The enzyme will not function without this cofactor present. N-acetylglutamate is synthesized from glutamate and acetyl-CoA, and when you ingest large quantities of protein in the fed state, you will have high levels of glutamate coming from the proteins. In addition, you will have high levels of acetyl-CoA coming from the metabolism of carbohydrates and fat, and thus you will have higher levels of N-acetylglutamate, and this will serve to activate the urea cycle. And this is the manner in which the urea cycle is regulated because, as I told you before, carbamoyl phosphate synthetase 1 catalyzes the rate limiting step, and therefore this activator can control the activity of the urea cycle as a whole. Now, don't confuse the enzyme carbamoyl phosphate synthetase 1 with the enzyme carbamoyl phosphate synthetase 2. Synthetase 2 is involved in the pathway of pyrimidine synthesis. So the molecule carbamoyl phosphate is also an intermediate in the synthesis of nucleotides that are pyrimidines, like thymidine and cytosine and uracil, and that pathway involves an enzyme called carbamoyl phosphate synthetase 2. Back to the urea cycle, the second reaction of the cycle also occurs in the mitochondria. In this reaction, a molecule of carbamoyl phosphate is combined with a molecule of a structure called ornithine to form a molecule called citrulline. The enzyme that catalyzes this step is called ornithine transcarbamylase, and as we will see in a couple of slides, deficiency of this enzyme is the most common metabolic defect of the urea cycle. Just to review the first two steps of the urea cycle that occur in the mitochondria, ammonia is combined with carbon dioxide to form carbamoyl phosphate. The enzyme that catalyzes this step is carbamoyl phosphate synthetase 1. The next step forms a molecule of citrulline. The enzyme that catalyzes this step is ornithine transcarbamylase. This step involves addition of a molecule of ornithine, which comes from the urea cycle in the cytosol and then enters the mitochondria to be combined into citrulline. Citrulline can then exit the mitochondria via a transporter to go through the next steps of the urea cycle that occur in the cytosol. On this slide, I've shown the remaining steps in the urea cycle that occur in the cytosol. Citrulline is converted into a large molecule called argininosuccinate. This step involves the amino acid aspartate. You may recall that I told you aspartate donates the second nitrogen to the final molecule of urea, and this is the step where aspartate is involved. Argininosuccinate can be converted into arginine. This step involves formation of a molecule of fumarate, and you may recall that fumarate is part of the TCA cycle, so this represents a crossroads between the urea cycle and the TCA cycle. From arginine, you can synthesize urea, and in addition, you regenerate ornithine. Recall that ornithine was consumed in the mitochondria. It is regenerated in this step, and it can cross back into the mitochondria to participate in the urea cycle again. Let me just point out to you that all the structures I've shown on the screen are amino acids. They all have an amino group and a carboxylic acid group. So citrulline is an amino acid, so is argininosuccinate, so is arginine, and so is ornithine. Only arginine is a standard amino acid encoded by our genome. The other three structures are non-standard amino acids. They can be synthesized as part of the urea cycle or other metabolic pathways, but they are not coded for by our genome. Along those lines, let me mention citrulline. It is a non-standard amino acid, as I said before, not encoded by our genome, but it can be incorporated into proteins via post-translational modification. And it turns out there is more incorporation of citrulline into proteins in settings of inflammation. And in the condition of rheumatoid arthritis, anti-citrulline antibodies are very helpful in the diagnosis of the condition. Molecules called anti-cyclic citrullinated peptide antibodies, or anti-CCPs, are positive in up to 80% of patients with rheumatoid arthritis, and those antibodies are directed against citrullinated structures which come from citrulline, the non-standard amino acid in the urea cycle. So the point of the urea cycle is to metabolize ammonia into urea. So if there is any disruption of the urea cycle, a condition called hyperammonemia will occur, where there are high levels of ammonia in the plasma. This is commonly seen in advanced liver disease. Recall that the liver is where the urea cycle occurs. So if the liver is sick, the urea cycle will not occur as it should. Rarely this can be caused in children who are born with deficiencies of one of the urea cycle enzymes. Those are called urea cycle disorders, and we'll talk more about those in a minute. On a biochemical level, the reason that high levels of ammonia are bad is because they can deplete alpha-ketoglutarate, which is an intermediate in the TCA cycle. Recall this step I've shown at the bottom of the screen in ammonia metabolism. Ammonia can be combined with alpha-ketoglutarate by the enzyme glutamate dehydrogenase. This ultimately leads to the production of glutamine. So if the ammonia levels are very, very high, all of the alpha-ketoglutarate will be tied up in glutamine, and you won't have enough for the TCA cycle so that cells can function properly. The main clinical effects of hyperammonemia that you will see when you examine patients have to do with the central nervous system. High levels of ammonia can lead to cerebral edema. They classically cause a tremor called asterixis. If you ask patients to hold their hands out in front of them and put their palms up like they are telling someone to stop, their palms will flap, and that is asterixis. You can Google this on YouTube to see examples of liver disease patients who have asterixis. Patients with high ammonia levels are often confused and have memory impairment. They can have slurred speech. Sometimes they can develop vomiting, and this can progress to coma and even death. One of the ways to treat patients with high ammonia levels is to place them on a low protein diet. If they don't consume steak and chicken and other sources of protein, there will be less ammonia in the bloodstream. Another way to treat them is to administer a laxative called lactulose. Lactulose is a synthetic disaccharide. It functions as a laxative by drawing fluid into the colon, but it's given in patients with hyperammonemia for a very different reason. There is colon breakdown of lactulose by bacteria into fatty acids, and this lowers the colonic pH. As a result, this favors formation of the positively charged NH4 molecule over NH3. The NH4 in the colon is not absorbed, and thus it is trapped in the colon, and as a result you will lower the plasma levels of ammonia by giving patients lactulose and converting any NH3 that finds its way into the colon into NH4, which will then be trapped and excreted in the stool. There are some other treatments for hyperammonemia that are generally only used in children who have urea cycle enzyme defects. These include ammonia detoxicants. These are substances like sodium phenylbutyrate, which can be given orally, or sodium phenylacetate and sodium benzoate, which can be given IV. These conjugate with glutamine, and as a result, the glutamine is excreted in the urine, and this results in the removal of nitrogen from the body and therefore less ammonia. In addition, children who have urea cycle defects are often supplemented with the amino acid arginine because the urea cycle disorders make arginine essential. It can no longer be derived from other structures in the urea cycle. Now let's talk about an ornithine transcarbamylase deficiency, or an OTC deficiency. This is the most common inborn error of metabolism in the urea cycle. It is an X-linked recessive condition, and children born with this enzyme deficiency have very high levels of carbamoyl phosphate and very high levels of ammonia. If we go back to this slide here, ornithine transcarbamylase is dysfunctional in an OTC deficiency. As a result, the level of carbamoyl phosphate will rise, and in addition, the level of ammonia will rise. An additional problem these children have is high levels of orotic acid in the plasma. Why is this the case? Well, remember that I told you that carbamoyl phosphate is also an intermediate in pyrimidine synthesis. In the pyrimidine synthesis pathways that lead to formation of cytosine and thymidine, carbamoyl phosphate is converted into orotic acid. So when the carbamoyl phosphate level gets very high from the urea cycle being dysfunctional, that will in turn lead to very high levels of orotic acid. OTC deficiencies present in infancy or childhood. The age at presentation depends on the severity of the defect. If the defect is severe, it will occur after the first few feedings when the baby begins consuming protein. The symptoms will be from hyperammonemia. These may include somnolence and poor feeding. It can include seizures. It can also include vomiting, lethargy, and even progress to a coma. For your board exams, make sure you don't confuse an OTC deficiency with another metabolic condition called orotic aciduria. Orotic aciduria is a disorder of pyrimidine synthesis, and just like an OTC deficiency, babies with orotic aciduria also have high levels of orotic acid. However, only in an OTC deficiency will you see high ammonia levels because an OTC deficiency involves the urea cycle. So the ammonia will lead to encephalopathy, and therefore the child will have lethargy and coma in an OTC deficiency. You will not see those findings in orotic aciduria. There is another urea cycle disorder called citrullinemia. This is a deficiency of the enzyme argininosuccinate synthase. It results in elevated citrulline levels, as the name implies. In addition, the levels of arginine are low, and there is hyperammonemia, as there is with any urea cycle defect. If we go back to our overview slide, argininosuccinate catalyzes this step right here, so this will be dysfunctional. As a result, the level of citrulline will get very high, and in addition, you will not synthesize the amino acid arginine, so arginine levels will get low, and then of course you're disrupting the urea cycle, so ammonia levels can build up. There are other urea cycle disorders. Deficiencies of each enzyme in the cycle have been described. All of them are autosomal recessive except for OTC deficiency, which, as I told you before, is X-linked recessive. They all lead to hyperammonemia. If you see a baby with hyperammonemia, you should think of a urea cycle disorder, and they all lead to buildup of various urea cycle intermediates depending on which enzyme is dysfunctional. And that concludes our module on ammonia.